Hello and welcome back, friends. Welcome to another video tutorial from Shomu's Biology. And in this lecture, we want to talk about the second chapter, and this is about the sexual reproduction in flowering plants. In the earlier classes, we've talked about the reproductive strategies that different organisms take, that we've seen asexual reproduction, and we've also seen the overall idea behind sexual reproduction. Now, we know that the sexual reproductive strategy between a plant and an animal is completely different, right? So, the sexual reproduction of plant is mostly concerned about the center event, that is the fertilization, which is true for animals as well. But the fertilization event for plants are different. It's known as a double fertilization, not single fertilization event, which is true for any other living animal that we know. So plants only undergo this double fertilization event. So right now we are concerned about only those plants which are flowering. We call them flowering plants or angiosperms. So we will be looking at the sexual reproduction in flowering plants or in angiosperms. And in this whole chapter, we'll talk about the three important sections. One is the pre-fertilization event. So all the events that occur before the double fertilization. And inside the pre-fertilization event, we mainly look at the morphology and the anatomical structures and the differences between the structure between the male part of the flower, the female part of the flower, and those structures. The male part is known as the stamen, the female part is known as the pistil. Apart from knowing the whole structure and how they exactly produce their component, which are the gametes, for any fertilization event, we've studied earlier that for this sexual reproduction, we need to produce gametes, which are half the number of chromosomes, somatic cell chromosome containing components or cells. In this case, if the somatic cell carries 2n number of chromosomes, so these cells will carry n number of chromosomes, which is true for animals as well as these sexually reproducing plants. So this male part will produce the male n number chromosome containing cell, which are known as pollen grains. You probably have seen pollen grains a lot of times in your life. That is the male part of a flower, equivalent to the sperm in case of the animal. And on the other hand, plants also need to produce the egg, right? And the egg is produced inside the specific structure in the plant. So egg and that pollen grain, both of those need to fuse with each other to form what is known as the zygote. So here we will also see the fusion of the male and female gametes, and then the fertilization event will take place. And then the third part of the lecture will be the post fertilization event, that is after the fertilization, after the double fertilization is done, then what happens to that fertilized zygote, that is 2n number chromosome containing cell, the first 2n number chromosome containing cell, and how exactly that cell give rise to the future generations, right? Because in plants we know all the informations that are needed for making a new plant is inside the seed. So exactly which part of that zygote turn into a seed and how exactly the whole process works, that's what we will see in this complete chapter. So in this first video, we just need to talk about the male part of this plant, okay, as well as we'll see the female parts. So let's begin with the pre-fertilization event and we'll look at the morphology of these male portions of the plant. So the male part of the plant is known as this stamen, right? Stamen, and we also call it the microsporangium, which is a part of the stamen. And similarly for females, it's pistil, and the megasporangium is a part of the ovule, which carries all the egg. So let's begin with the discussion about stamen. So the stamen, for all the different plants, we studied earlier in the last class that there are few plants who only have flowers with stamen, and there are few plants who only have flowers with only pistil. We call them staminate and pistillate plants. Now those are unisexual plants, that they only carry one type of sex, either male or female, right? But apart from that, there are also many plants out there who have both the stamen and pistil region in the flower, in the same flower. Okay, so let's look at the structure of the stamen. So if I draw the structure of the stamen, it carries two separate regions. Remember, one is structure like this, known as the anther. Another one is this filament. This is anther. So anther and filament, both the structures. And if you look at the anther, there are two sections to it. So it is bilobed. Bilobed. Bi means two. So it divides into two different portions. And also in this bilobed structure, they are also having two separate layers of tissue inside, known as theca. So two separate theca is present and they are bilobed in nature. That is the complete structure of male part of a flower. This part anther, this part filament, and all these structures together is known as the stamen. Okay. Now remember one simple thing. Whenever we are looking at the male part of a flower or female part of a flower, before understanding the fertilization event, you need to know the morphology in details and how exactly the gamete is produced. That's what our idea is. That's what we need to find out in this particular video. So in this complete structure, this filament is only a structural component that holds the anther. All the major processes going on inside this anther near the theca, because this anther is the place where they have a complex tissue arrangement that will help to produce the pollen grains. So ultimately, what is the gamete? What is the male gamete for the plant? The pollen grains. So whenever pollen grains are produced, that pollen grain can now further germinate and can go through the female part, which is pistil, and cause the fertilization event to occur. Okay, so from perspective of a plant, we need to develop the pollen grain. But how exactly the pollen grain is synthesized? To understand that, we need to go inside the anther. Now if you go inside the anther, you cut the anther, let's say a cross section of the anther. How it will look like? It will look something like this. Let me draw it. It is like 4 separate sections like this, something like this. 4 separate sections are out there in this cross section of the anther. And you also see that this membrane that we're looking, it's not a single membrane. There are multiple layers. Inner and outer layers are out there. Okay, multiple layers are out there, and these layers are forming this specific structure of the anther. So try to imagine a cross section, and you are looking at a top view from the cross section. Then we see something like this, and the structure looks like they have these 4 separate sac-like structures. These sac-like structures are known as microsporangium. So these sac-like regions are the place where the process of male gamete production will happen. Okay, so this microsporangium, they're surrounded by all these different structures out there, and they make this fold, like as I told you, like it's a bilobed, because there are 2 fragments. If you look at the anther, you'll see like it's lobed. That lobe is generally formed due to this 3 layers, and the layers invaginating inside to create these lobes. Now these are the places where they will produce those microspores. Now remember one simple thing, microspores are not present from beginning. Microspores are the preliminary part of the pollen grain. Okay, and microspore means they are kind of the n number containing cell. So they are you can say microspores as gametes, the male gametes for the flowering plant, but they are not present from before. So normally here, instead of microspores, what we have are simply layers of tissues in all these 4 regions, layers of tissues, and those tissues are known as what? Sporogenous tissue. Sporogenous tissue. Another interesting fact that I should tell you is that this structure of anther, if you look closely, they carry this 4 separate layers actually, 4 separate layers. See, 3 of the layers are drawn here with black color, and the one layer, the innermost layer, is drawn with this red color. So you see the outermost layers, they start folding and making this kind of lobular structures, and you can easily see this line formed due to the formation of this lobe. This is known as line of dehiscence. ENC, line of dehiscence. Okay, this the structure is formed with the outer 3 layers of this anther structure, or you can say outer 3 layers of this microsporangium. And we know the innermost layer is right now in contact to the sporogenous tissue. So this innermost layer, which is pictured here with this red color, is known as tapetum. Tapetum. Now this line of dehiscence and tapetum, they have their unique function. This line of dehiscence is required to the future process of spore release or release of pollen grains. When the microspores mature, this line of dehiscence helps them to be dispersed. And you know, dispersal of the spores are very much required, because after the dispersal of the spore only, the spore can contact the female portion of the flower, right? Which is known as the stigma of the flower. We'll talk about that in future. But this tapetum layer, which is the innermost layer, this tapetum is in contact with sporangium tissue, and tapetum actually nourishes and provides the nutrients to the sporogenous tissue so that sporogenous tissue can grow, right? Now this sporogenous tissue has specific type of cells that can give rise to microspores, right? So imagine this known as the, so all these tissues and the cells that are present there are known as mother pollen cell or pollen mother cell, PMC. So those cells known as the pollen mother cells. Why pollen mother cells? Because those cells can give rise to the formation of pollen. So they are mother for the formation of the pollen. That's why called as a pollen mother cells, which is a type of sporogenous tissue. So pollen mother cells are present in the sporogenous tissue, getting the nourishment from tapetum layer, and they start growing. So once they get all the nutrients, proper nutrients for their growth and maturation, then what happens? These pollen mother cells, which are, so let me write it here, the pollen mother cells normally is 2n cell. That means it carry 2n number of chromosomes in the nucleus. But these pollen mother cells are undergoing meiosis cell division. And you know what happens to meiosis cell division? We just saw that in the last class, that meiosis cell division changes the number of chromosomes by half in the daughter cells. So if the mother cells have 2n number of chromosomes, and the daughter cell will have only n number of chromosomes. So this meiosis cell division of pollen mother cells give rise to the formation of microspores. And the microspores that are formed, I told you microspores are the actual gametes because they carry only the n number of chromosome, which is half of the somatic cell chromosome. So these are the gametes, and we know gametes always contain n number of chromosome because two gametes will fuse. So if both of them are n number of chromosomes, after the fusion they become 2 number chromosome containing cell. So these microspores are produced after the conversion from PMC by meiosis cell division, and this process is known as microsporogenesis. Microsporogenesis is a process with which pollen mother cell converted into the microspore. Now once microspore is produced, the microspores start to present here. Let me draw the microspore. Let's say these are the microspores which are produced there. Okay. Now the microspore is not ready to be released from this microsporangium. It's not ready to be released from microsporangium because the microspores produced are not dry enough. Because in plants, the idea is to release the spores outside into the air. And you know, many a times the spore needs to land to the stigma of the female part of the flower. For that they need to be dry, they need to be very lightweight. So most of them are lightweight, but they need to be dry to become very lightweight. So dehydration of these microspores are required. And once dehydration takes place for these microspores, then only they are able to be transferred from this microsporangium and the structures of microsporangium outside, and that dispersal is favored by line of dehiscence formed by these three outer layer of the microsporangium, right? That's what happens. So the dehydration is very important. Once microspores are dehydrated, they are converted to pollen grains. Pollen grains. They form pollen grains. So these pollen grains that we know, like if you take a flower from outside, if you take it and if you look at it, you'll see, like if you just take the China rose in your hand and you simply take it in your hand, and you'll see the anther part. If you rub it in your finger, you'll see that yellow dotted things that will come into your hand. That are the pollen grains, right? That pollen grains are dry, and it's a converted form, a modified version of microspore. Pollen grains are also the same in number carrying cell, but microspore, and they will ultimately migrate either in between the same plant or to the other plants. Okay, they can migrate. So that's the idea of forming pollen grain. So now once the pollen grain is formed, the structure of pollen grain is also a little bit different, because you know, when the microspore is formed, the microspores are cells. Like now the cells have differences in their structure, and especially if you look at a pollen grain has two separate layers, inner layer and outer layer. So now let's look at the structure of pollen grains. Okay, so pollen grains has a simpler structure with two separate layers, inner layer and outer layer. Outer layer is known as exine and inner layer intine. Exine and intine. Now exine contains a specific molecule, okay, that is known as sporopollenin. The name is sporopollenin. This sporopollenin is known as the toughest material known. Actually not toughest, it's wrong thing to term, but sporopollenin is known as the most resistant compound that human know till this point, because you know, it's very much resistant to high temperature, resistant to the change in pH, because if you put it into acid or alkali, nothing happens, because under the intine layer inside there is the cytosol, and there is where the nucleus is found. So the inner side of this pollen grain is always living, but the exine layer of this pollen grain, known as the made with the sporopollenin, is really tough, and it helps the pollen grain to be protected, because you know, the pollen grain that is produced by a plant needs to travel a lot of distance, right? It needs to travel through air, or some cases through water. It needs to be disposed into the acidic environment, basic environment, doesn't matter, because they carry this sporopollenin. Now this cell inside, they can divide into two different types of cell: vegetative cell and generative cell. So once this pollen grain is ready to be dispersed, now the cells start dividing. Even before dispersal, they need to be readied for dispersal, and they need to divide into two types. What are the types of the cell? This cell inside divides into two, one carrying most of the part of the cytosol, okay, known as the vegetative, okay, and the other small section of the cell that remains there. So vegetative cell contains a large section, and the nucleus that is present inside is also huge. It's big. Vegetative cell that is huge and big out there, and the smaller sections, it carry very dense nucleus in the center, while the vegetative cell don't have a dense nucleus. It has like an amorphous structure of this nucleus out there. So this large cell is vegetative, and the small cell with dense nucleus is known as generative cell. It's known as generative cells. Okay. Now most of the plants, contain these two types of cell. Almost 60% of the plants before the dispersal of the pollen grain produce this two type of cell, vegetative cell and generative cell in the pollen grain. And very few type of plants are out there, almost like 30% of the plants out there. They also have a division of this vegetative cell further to produce this generative cell further to produce two separate generative cells, because remember the vegetative cell is not involved in the process of actual fertilization. Generative cell is the one which involved in the part of fertilization. So generative cell needs to be duplicated. So few plants duplicate, most of the plants duplicate this generative cell and make two versions of this male plant gamete just before fertilization event, after landing into the stigma of the female part of the flower. But 30% of the plants are out there who already convert and duplicate this generative cell so that they can initiate it the moment it's in contact with the female part of the flower. Okay, that in a sense are the different components of the plant, of the male portion of a plant. Okay, now we will see the structure of pistil, that is the female part of the plant and its structure, and we'll see then, after knowing both the structures, we are going to see how exactly the double fertilization event take place.